First of all, really, it's an incredible honor and a privilege for me to be bestowed with the H.K. Firodia Memorial Award. Not only the illustrious awardees that have gone by, but the fact that it's been awarded for science and the pure pursuit of science is something that really is quite extraordinary. I'm really deeply honored, humbled that a eminent industrialist a doyen of Indian industry would take the effort to institute an honor for the pursuit of something that is a pleasure to pursue. And in fact, I'm very grateful. So a couple of clarifications. I'm the center director of the National Center for Biological Sciences, which happens to be a center of the Tata Institute for Fundamental Research. So this evening, we have a clean sweep. The TIFR director, Sandeep, and me are both here on the stage. And it's really a pleasure for me to be speaking in front of colleagues. What I thought I'd do is speak to you about how I'm seeing the changing landscape and the changing language of biological systems, and somehow convince our younger generation that there are incredible opportunities in this change that we are undergoing. So without any further ado, I'm going to get to the meat of this 20-minute talk. So biology, as you all may or may not know, is the science of this century. Our future depends on how we understand all life forms on this earth, from the I shouldn't say lowly, from the incredible bacterium to the nematode, to various plants and animals, and some incredible human beings. They're all biological creatures. But the question that many of us ask, what makes biology unique? And this is a question that was posed many years ago and is still continuing to be posed. And I think today we have a very, very interesting way of addressing what biological systems are and how they are unique. And I want to take you through some of the understanding that we've come through and why it's such an exciting time to be doing biology, especially for the younger people in the audience, and in fact, even for the older people to just think about how biology is unique. So, let me, before I get to what we think about the language of biology today, let me give you a bit of historical context. And one big part of this history is that nothing in biology makes sense except in the light of evolution. Two stalwarts up here, Charles Darwin and Gregor Mendel, thinking and working at the same time, but they never actually managed to correspond with each other. One was talking about an explanation for all the different life forms we have on the planet, and the other, the explanation of heritable traits. It's extraordinary that these two stalwarts of biology were in fact working and thinking at the same time, but their paths didn't cross, except as a little book that Gregor Mendel had sent Charles Darwin, which it turns out lay unopened on Darwin's bookshelf. And had he perhaps read that book, he would have even understood the means by which the evolutionary change that he was talking about could have come about. Today, that problem would not arise because we have the internet, and he would have sent him an email and figured it out. But let me just say that from a descriptive science in the early 1900s, biology started at this time as well with a few driving concepts. The theory of evolution, as I just mentioned, the cell theory, and the laws of genetics. All of these have provided an overarching explanation for the existence of all life forms on this planet, and they still continue to do so. These ideas, if you will, were a largely European adventure. Darwin and Mendel, and as I mentioned, they had tremendous contrasts in styles. But there were exceptions to this kind of thinking of a descriptive explanation, and that of Darcy Thompson, who brought in the idea that you could bring in physical principles to understand biology. And then our own Jagdish Chandra Bose, he explored with great abandon the physical properties of living materials. And some of his extraordinary work in understanding how muscles twitch and the electrical properties of different sorts of materials, living materials, was something that he explored, and we today acknowledge that exploration. Then in the 1900s, little later on, from 1950s, I'd say, predominantly to the 2000s, biology became much more sort of chemistry-oriented. It became reductionist and mechanistic with the discovery of the stuff of hereditary DNA. We had a virtual molecular biology revolution. Biology was being translated into molecules and what sorts of molecules existed in biological systems. Watson and Crick had discovered the structure of the biological system. Luria, Lederman, Delbruck, these are some of the individuals at that time who were driving thinking in the biological context. This again, except for perhaps Watson and Crick working at LMB, the Laboratory of Molecular Biology in Cambridge, was a largely American kind of revolution. This culminated, if you will, in a huge new scope for biology. But while this was going on, in India we also had a tremendous thinker and molecular biologist, perhaps one of the few people who should have been recognized with one of the top prizes in biology, G.N. Ramchandran. In fact, it was on his work on the triple helix that much of the understanding of the X-ray structures of the DNA were decoded. In the contemporary context, we have today, in just at the turn of this century, 2000, the human genome was decoded. And today, in the past 15 years, not only has the genome been decoded, but we hear about scientists being able to edit genomes of not only organisms, but humans. Raising, I think, very important questions that I think ethicists of our time should be engaging with, and in fact, this kind of exploding of understanding and possibilities of changing the DNA structures, in many ways, is going to be an important question we're all going to have to grapple with. The understanding of editing of DNA, by the way, I should mention, came from scientists wanting to understand how bacteria evade their own viruses. So the gene editing technology, the CRISPR technology of today, came from some scientists sitting on a beach in Spain exploring how the bacteriophages are being repelled by bacteria. And then, of course, the new technology of gene editing emerges from that search. But more in the chemistry context, this is, in some ways, the structural era of biology. And here, one of our own, in fact, one of products from the MS University in Baroda, Venki Ramakrishnan, who then went off to America, and then ended up with the Nobel Prize in Chemistry in 2009. also honored in these awards. He ushered in a tremendous revolution in structural biology. His work it was able to show at atomic detail the structure of perhaps the largest living machine that we have in our cells, the ribosome. In fact, because from the work of Venki, Ada, Jonath, and Tom Steitz, was able to show how every hydrogen atom in this machine is organized and located, and how this contributes to its function. So how proteins get made inside a cell, reading the instructions from the genome was a great achievement that Venki put together along with some other colleagues. In parallel, we've had in the last, I'd say in the last 8 years, the biggest revolution in imaging. Today, we're able to look in a living cell at maybe 10 times atomic resolution, at a nanometer to a few nanometers resolution. And we're able to see inside a cell structures that were unimaginable in just 1999. So there's been a tremendous transformation of technology. And in fact, here I'd say is something that's, if I can get my, here we go. If I were better at playing video games, I would manage this. Here we go. So that's a structure that's being pinched off from the surface of the cell, observed at 10 nanometer resolution. It's really extraordinary. And this can also now happen in Bangalore, where we also have a cryo-EM facility for looking at exactly the same kind of technology Venki uses in Cambridge, and generating an incredible amount of data. So my colleagues in Bangalore, Vinod Kumar and Das, at NCBS and INSTEM respectively, they have been isolating ribosomes from different organisms and finding things that Venki also would not have dreamed of. And this is just in the pace of 5 years. They've discovered structures of the ribosome and how ribosomes stutter and stall when they translate the information from the genome in ways that we wouldn't have thought possible unless we'd observed deeply into these structures. But all this has meant generating huge amounts of data, terabytes of data. And this, I should say, has led us to what we now call in biology the big data problem. The big data is everywhere in biology. It's the human genome is enormous amount of data, but the human genome produces a huge deal of omic data. It produces the transcripts, it produces proteins, it produces the epigenome, it produces lipids, it produces all kinds of information. And this information just grows arithmetically, and it explodes in a way that makes us sort of almost balk at the quantum of such information. And if we then take that information and bring it into the clinic and multiply it by, say, the number of human beings on this planet, that's the sort of information we are looking at right now. So it's an incredible task. Big data is in every aspect of biology today. And one can look at this as a problem, or one can look at this as an opportunity. And I think my young friends, especially in the audience, and I think perhaps Nandan might be talking about some of this later, there is an incredible opportunity here about engaging with this big data. So the big data needs to be crunched into some kind of a funnel so that we can begin to understand it. And this big funnel is turning out to be algorithmic understanding of information. This big data, and in fact, one example came up very recently where one of the people who actually ended up decoding the human genome, Craig Venter, he set himself a challenge to see whether you could take people's faces and ask with a few traits connected to these faces and genomic information, see whether one could in fact, from genomic information, infer the faces of faces that had not been fed into this big data funnel. Simply said, if you look at the next slide, what you will see is that the faces that were able to be decoded. So here is a face on the left, which is the real face, and the face that is being read from genomic information that's fed into this, into the algorithms created to crunch this big data, is the face on the right. This extraordinary similarity, and this is an incredibly controversial area right now, and some people even doubt the veracity of this information. But I'm just to say that the fact that one could even conceive of doing this would not have been something we would have done a few years ago. So, and this is because of the fact that we can combine big data and artificial intelligence. Big data and artificial intelligence together contribute for us to make these correlations which would otherwise be impossible. And I think therein lies the great possibility ahead. But I must say that the fact that we combine big data and artificial intelligence doesn't mean we actually understand what's going on. We don't understand how that genome that is connected to that face actually contributes to the building of that face. We don't understand the chemistry, the physics, or the biology in that process. And some people would say that we will never understand that. We will just have these correlations that we can make. And some people argue that biology is unique, and we perhaps will never understand even how a cell works. But I must say, even that is being challenged today. The natural sciences, physical, chemical, and mathematical, are coming together in an extraordinarily exciting journey and creating a new language which allows us to look at biology today. I mean, the one distinction between the physical sciences and the natural sciences is that biology has a history and a contingent future, which is something that, perhaps Sandeep might allude to when he talks about the universe. But it's something that in biology we take for granted. So there is no type individual. There are individuals in a population. And so how does one take a gene and convert that into a singular understanding? And that's, I think, the big challenge of biology today. But I must say that at the level of a cell, there's a completely new perspective that's emerging. And this is because of the integration of the physical sciences with biology. So at one end, we have the molecule and our detailed understanding of the molecular structure. As I said, now we can look at these molecules even in living cells and get a very detailed, almost close to atomic resolution of structures in living cells. And on the other hand, we have big data and AI. But incrementally, we are building an understanding that connects these molecules to the larger scale structures that are being built up inside a cell. We're connecting molecules and the way they build structures and architectures, for example, filaments inside a cell, and for example, how organelles inside a cell, how the nucleus, how endosomes, how mitochondria gets constructed, and then how a cell builds itself, how it builds a moving machine. And all this is happening because we can take, at least at this scale, at the scale of molecules building structures in a living system, completely driven out of equilibrium because all these systems are consuming energy. We can build structures from that we call at the mesoscale, at the scale of microns, and understand these structures completely. We have theories that explain how these structures get put together and experiments that can contribute to them and bring in all our understanding of the natural sciences that is available in front of us, from condensed matter physics to applied mathematics and everything in between, including the engineering sciences. So all this put together brings us an extraordinarily first principles understanding of the stuff of a living cell. It's also the integration of the physical sciences with biology and the availability today of stem cell-based technology that allows us to experiment with how things get put together when you connect the physical, chemical, and mechanical inputs with the molecular and information that's encoded in the gene. And this is, I can't think of a more exciting opportunity for young people to consider that this may be how one might study a living system. There is a new language for biology, and it's all the languages of science that come together to study the biological system as a system that can be studied on its own. So, of course, there is the big data. There is AI, which allows us to assimilate this information. But it then needs the rest of the sciences to be able to understand what is going on. So here's a picture. In fact, I'm going to leave you a picture of a cell that's trying to swallow its prey. It's a cell from your immune system. It's a neutrophil, which is running after a staphylococcal bacterium. It's in sort of a blood smear. So here's the bacterium, and the neutrophil will start its journey now, and it'll go and find this bacterium. The bacterium is actually excreting something, so it can actually sense the bacteria. And at the end of this movie, what you will see is that the cell actually ends up, that's suspense. Whoops, there you go. It finished the bacterium, it goes off, and then it burps. So the burp was in the sound on this movie, which I didn't play. Yeah, so I just want to end here by also, one second, did I get the next slide? Sorry. By thanking you all this evening for listening so patiently, thanking the H.K. Firodia Memorial Foundation and their wonderful expose of this kind of science, and also trying to bring science and innovation together. But I want to thank my colleagues, Obed and Vijay, who are former H.K. Firodia awardees, but they were instrumental in my being here today. Krishnan, another Satyajit, Veronica, and of course Madan, who you saw. And none of the work that I do in my lab, which I will have to present to you on another occasion, because I didn't talk about work that comes from my lab, but none of this would have been possible without my fabulous students and postdocs who have worked with me all these years. Thank you so much for listening.